Hi, I'm Castigador, and together with my group mates, we will explain bacterial conjugation. Let's begin with defining bacteria. Bacteria are microscopic, single-celled organisms that thrive in diverse environments. They can live within soil, in the ocean, and inside the human gut. Human relationship with bacteria is complex. Sometimes, they lend a helping hand by curdling milk into yogurt or helping with their digestion. At other times, they may be destructive, causing diseases like pneumonia and MRSA, to name some. These bacteria transfer DNA by direct cell-to-cell contact if a conjugative plasmid is present, and this is what we call bacterial conjugation. An example of a conjugative plasmid is the F factor, which is best studied. Many of these F factors direct the formation of sex pili, which attach the donor cell that contains the F plasmid, called the F positive cell, to an F negative cell, which is the recipient. Also, the F factor has several IS elements that aid plasmid integration into the host cell's chromosome, making the F factor an episome that could exist out of the bacterial chromosome or be integrated into it. There are three types of bacterial conjugation. The first being the F positive and F negative mating. The second is HFR conjugation. And the third being F prime conjugation. The first type of bacterial conjugation is the F positive and F negative mating. In 1952, William Hayes demonstrated that the gene transfer observed by Lederberg and Tatum was unidirectional. That meant that there were definite donor strains and gene transfer was non-reciprocal. He also found out that in F positive and F negative mating, the progeny were only rarely changed with regard to auxotrophy. However, the F negative strains frequently became F positive strains. Now that these results were studied, we can now understand and readily explain that the F positive strain contains an extra chromosomal F factor carrying the genes for sex pilus formation and plasmid transfer. Once contact is made, the pilus retracts, bringing the cells into close physical contact. The F positive cell prepares for DNA transfer by assembling the type 4 secretion apparatus using many of the same genes used for sex pilus biogenesis. The sex pilus embedded into the secretion structure. The F factor then replicates by rolling circle replication. During conjugation, rolling circle replication is initiated by the relaxosome, which are encoded by the F factor. The relaxosome nicks one strand of the F factor at a site called ORIT. Relaxase remains attached to the 5' end of the nicked strand. As F factor is replicated, the displaced strand and the attached relaxase enzyme move through the type 4 secretion system to the recipient cell. Dividing plasmid transfer, the entering strand is copied to produce double-stranded DNA. When this is completed, the F negative recipient becomes F positive. The second type of bacterial conjugation is called the HFR conjugation. Remember that by definition, the F positive cell has the F factor free from the chromosome, so in an F positive and F negative mating, chromosomal DNA is not transferred. However, within this population, a few cells have the F plasmid integrated into their chromosomes. This explains why not long after the discovery of F positive and F negative mating, a second type of F factor mediated conjugation was discovered. In this type of conjugation, the donor transfers chromosomal genes with great efficiency but does not change the recipient bacteria into F positive cells. Because of the high frequency of recombinants produced by this mating, it is referred to as HFR conjugation, and the donor is called an HFR strain. HFR strains contain the F factor integrated into their chromosome rather than free in the cytoplasm. When integrated, the F plasmid's TRA operon is still functional. The plasmid can direct the synthesis of pili, carry out rolling circle replication, and transfer genetic material to an F-negative recipient cell. However, rather than transferring just itself, the F factor also directs the transfer of host chromosome. DNA transfer begins when the integrated F factor is nicked at ORIT. As it is replicated, the F factor begins to move into the recipient. Initially, only part of the F factor is transferred, followed by the donor's chromosome. If the cells remain connected, the entire chromosome with the rest of the integrated F factor will be transferred. This takes about 100 minutes to accomplish. However, the connection between the cells usually breaks before this process is finished. Thus, a complete F factor is rarely transferred and the recipient remains F-negative. When an HFR strain participates in conjugation, bacterial genes are transferred to the recipient in either a clockwise or a counterclockwise direction around the circular chromosome, depending on the orientation of the integrated F factor. After the replicated donor chromosome enters the recipient cell, it may be degraded or incorporated to the F-negative genome by recombination. The third type of bacterial conjugation is called the F-prime conjugation. Because the F plasmid is an episome, it can leave the bacterial chromosome and resume status as an autonomous F factor. Sometimes, during excision, an error occurs and a portion of the chromosome is excised, becoming a part of the F plasmid. A cell containing an F prime plasmid retains all of its genes, although some of them are on the plasmid. It mates only with an F-negative recipient, and an F-positive and F-negative conjugation is similar to an F-positive and F-negative mating. Once again, the plasmid is transferred as it is copied by rolling circle replication. Bacterial genes on the chromosome are not transferred, but bacterial genes on the F prime plasmid are transferred. These genes don't need to be incorporated into the recipient chromosome to be expressed. The recipient becomes F prime and is partially diploid because the same bacterial genes present on the F prime plasmid are also found on the recipient's chromosome. This way, specific bacterial genes may spread rapidly throughout the bacterial population. Although most research on conjugation in plasmids have been done using E. coli and other gram-negative bacteria, conjugative plasmids are also present in gram-positive bacteria belonging to Bacillus, Streptococcus, Enterococcus, Staphylococcus, and Streptomyces. Although there are also experiments done on gram-positive bacteria, much less is known about these systems. It seems that although transfer genes are also involved, there are fewer, and that it's possibly because a sex pilus may not be required for plasmid transfer. So that was bacterial conjugation. Thank you for watching our video, and see you next time!